Good afternoon, everyone. So I want to take this opportunity to welcome us to this very exciting session. My name is Irene Mukoi. I come from Kenya, and I work with the Ministry of Health as the Deputy Head of the HIV Control Program. I want to welcome us to this session, which I can see the room is already getting full, which means we are going to have a nice time for the session. Unfortunately, we won't have one of our speakers, so good for you because we can have a little more time for discussion and to get an opportunity to interact with our speakers. So the session is talking about next generation HIV prevention. I think just looking at all the sessions that have been going around HIV prevention and about pre-exposure prophylaxis, very interesting themes that are coming up. A lot of countries are trying to introduce PrEP now, and we have questions that are coming up around the best models of care, about the effectiveness of PrEP away from clinical studies, and when it gets into the real-world setting, around retention. People don't really know how to define retention, so we hope we get an opportunity to discuss about some of those things. And then in the next few months and in the next few years, we are going to have to think about the impact of pre-exposure prophylaxis in the context of all the combination prevention interventions that we know. And then part of our session is going to be talking about the future. So thinking around the introduction of newer prevention products, longer acting products for HIV prevention. And I guess we have to take this opportunity to discuss based on the lessons that we have learned from introduction of other prevention interventions. What is it that we need to think about now? What is it that we need to prepare about now ahead of the products being ready for the market? But we also have lessons that are learned from the hottest discussion that's been happening around about dolutegravir. So some of the prevention interventions we are thinking about safety concerns right from the beginning, depending on who we are targeting for our prevention interventions. So we look forward to an interactive, interesting session. And I want to take this opportunity to welcome Beatrice, who is my co-chair, to introduce herself and also welcome the first speaker for the session.
Hi, good afternoon. I'm Beatrice Greenstein, and I'm an infectious disease physician and researcher from Rio de Janeiro, Brazil. It's my great pleasure to introduce you our first speaker, who is Dr. Jared Beaton. Jared is the vice chair and professor in the Department of Global Health at the University of Washington, where he's also professor of medicine and epidemiology. He led seminal studies in prevention, the Partners PrEP study and MTN 020, the Aspire study, randomized trials that demonstrated the efficacy of oral tenofovir-based PrEP and the dapivirine vaginal ring for protection against HIV acquisition. Jared directs the University of Washington Fred Hutch Center for AIDS Research and is the co-principal investigator of the NIH-funded Microbicide Trials Network. He will talk to us about what do we know about PrEP from efficacy to effectiveness.
Good afternoon, everybody. Everything always sounds better in Brazilian Portuguese. I said when Beatrice was talking before we started. So it's great to be here this afternoon. It's great to have this opportunity to talk a little bit about some history of PrEP. And as Beatrice said, the title that I was given was From Efficacy to Effectiveness, and I modified that a bit and added to impact, because that is where we are beginning to see effects of PrEP. So, some disclosures. And an acknowledgement that the results of PrEP research, which we hear about today, rests on tremendous volunteerism, on really tens of thousands of people now at risk for and living with HIV. So in the beginning, there was no PrEP, and then there was PrEP. And it really does. There does seem that there was this moment when it was not discussed at all, and then it came. And then it is standing room only at poster discussion sections that are happening over lunchtime at this meeting. And I can very much remember PrEP when there when there were 10 people at PrEP sessions, and now they are bursting at the seams. So the interest is great. Whether that's translating into what we want it to be, which is fewer infections, is really the big question. So what we know about PrEP in terms of efficacy, effectiveness, and impact. And I'm framing this around efficacy, effectiveness, and impact. We learned about efficacy in clinical trials. There were many. And I think we will never again have such tremendous diversity in establishment clinical trials. More than a dozen studies that formed the regulatory package and the evidence, the WHO normative guidance evidence package for PrEP across countries and continents with really tens of thousands of persons and across every different type of exposure for HIV that is of great importance. And as we know, there were a lot of trials that had a lot of different answers, and we do not need to review again the confusion of those answers, but what we learned from those trials is that adherence is related to efficacy. And if there's low adherence, there's no protection. If there's modest adherence, there's more. And it gets higher as there's better and greater adherence within the trials, pointing to what we think is true, is that when adherence is excellent, protection is excellent. And of course, we saw that in the 2 trials that were done after the evidence of PrEP was established. So the 2 trials, IPERGAY and PROUD, that were initiated once PrEP had been demonstrated to be efficacious for HIV prevention, demonstrated nearly complete adherence and nearly complete HIV protection. At the individual level, we know this as well, out of the clinical trials and now into practice, of course, as well, that we find that people who use PrEP are nearly completely protected from HIV. And that is across populations, both women and men and others, as well as, and with numbers that point towards nearly complete protection, nearly 100% protection. Our introducers today mentioned safety, and I included information on safety because the safety of HIV treatment has been a discussion point, of course, in the last few months, and the safety for PrEP, we talk about it much less, but it was really a co-motivator of every one of the establishment clinical trials of PrEP because the bar for safety for a preventive medication is extraordinarily high because the tolerance of risk is very low for individuals who are otherwise healthy and don't have a disease that you may be protecting them from. Tenofovir-based PrEP demonstrated extraordinary safety, was well tolerated without significant renal or other toxicity, which was what was explored in the clinical trials, and very, very rare antiviral resistance. Importantly, and this I called out in a separate slide, there have been an intentional series of studies done to understand PrEP across the reproductive life cycle in women, demonstrating safety in the conception or safer conception period, throughout pregnancy, into lactation, as well as in women who choose not to become pregnant with no adverse interactions with contraception. As we think about new prevention products, we will need to be thinking about this as well, especially for where the epidemic is dominant, where women want to become pregnant or accidentally become pregnant. Demonstrating safety is not an option, and not an optional activity. So on the basis of this, of course, there was regulatory approval. Regulatory approval, which was really a tremendous motivator for PrEP, even for a drug that was already established and licensed. The regulatory piece was a tremendous motivator for people to use it. And there's a great quote here from Debbie Bernkrant at the FDA. And of course, now regulatory status is at nearly 4 countries worldwide, regulatory approval, and across the globe. So efficacy shows us that PrEP works. There you go. Lots of money spent, PrEP works. Lots of work. What we want to do, of course, is move from efficacy to seeing what it looks like in real world. And the effectiveness work on PrEP was done through demonstration studies and open label extensions that followed on the clinical trials. And this is tremendously surprising. So for those of us who do global health work and implementation science work, we expect that the efficacy measured in clinical trials will be the best you can ever get. And that effectiveness in real-world settings will always be less. And PrEP has been the opposite. Tremendous shocker. So on the left here are the results of the PROUD trial and the Partners Demonstration Project as 2 examples of the many demonstration projects that were done of PrEP, showing very, very high HIV protection in the context of very, very high adherence, of course, compared to the similar populations from the clinical trials themselves, IPREX and Partners PrEP. And in both cases, both adherence and effectiveness degree of HIV protection are considerably higher. people were adherent to PrEP when they knew it was safe for them and they knew it would work. And this is something that this is a story that carries forward not only in these demonstration settings, but now, of course, in practice. And of course, one of the first pivotal studies of that was done from the United States, from Kaiser Permanente. This is their early data, and this is carried through in larger data subsequently in the context of extraordinarily high HIV risk because of tremendous, gauged through very high STD incidence. HIV incidence was 0. So individuals who want PrEP take it and use it well in practice settings, so in demonstration projects and then into practice settings. And we see this is across all different populations now. We see this story repeated over and over again. So in blue on the left is in men who have sex with men, showing the HIV incidence in the placebo group of the IPREX clinical trial, and then falling to the PrEP arm of the IPREX trial, and then the open label extension, then the demonstration project, and finally at the bottom was 0 infections in those who were adherent. This same scooping downwards repeats itself in persons who inject drugs across placebo, PrEP, open label extension, and those who are adherent in serodiscordant couples and other heterosexual populations in red. And most recently, for the dapivirine vaginal ring, the same story is repeating itself in placebo to active to open label to adherent. Adherence was the Achilles heel of the clinical trials of PrEP, and of course, many millions of dollars and millions of hours of worry were spent understanding that if you don't take medicines, they don't work. But now we know that people do take them in real-world settings. This is one example from the United States, from one of the first demonstration projects. And what's here in the light blue at the top is across 48 weeks of PrEP follow-up, detection of PrEP levels detection of PrEP in blood samples at levels that were protective against HIV. And what is seen here is tremendously consistent adherence, that people who were returning to pick up their bottles of PrEP in an implementation setting were continuing to take it and take it well. Across the bottom, in the dark blue, are individuals who did not return for visits, and we're going to come back to this later when we think about retention into PrEP. But the basic story has been that individuals who are committed to taking PrEP and show back up for bottle refills take it has something to do with habit formation. You know if you are a flosser or not. You can raise your hand. Anybody want to raise their hand? Okay. People take PrEP, but why do people take PrEP? And one of the great things that I've loved about working on PrEP is thinking about why people take PrEP. And no one has ever said to me that they take PrEP because of a p-value or a New England Journal paper. People take PrEP because it does something in here. It decreases anxiety or makes them feel stronger themselves. It increases communication, disclosure, and trust with partners, or it makes sex better. And for a 30-something or 20-something, whatever the age of these guys are here, gay man in the United States, or a serodiscordant couple in Kenya, this is advertising from Kenya, or a 19-year-old woman in Cape Town, none of whom have ever lived in a world without HIV, to have sex without fear is extraordinarily powerful and has nothing to do with p-values. So the effectiveness of PrEP is clear, and PrEP is workable. So PrEP works and PrEP is workable. So let's talk about impact, because this is what we're in this for. We're in this to have an impact on the epidemic. An impact is being seen in studies of implementation and scale-up. This all begins in many ways with normative guidance. And the WHO boldly went out in 2015 and recommended PrEP as an additional prevention choice for individuals at risk for HIV globally. Of course, initial uptake was not great, and I'm an American, and of course I get the New Yorker, like many people, and this is great to see an article in 2013 on something that I care a lot about: why is no one taking PrEP? And this was the story. This was the story. It still is in many places the story. Why is no one taking the first medication to prevent HIV? That has begun to change. It's interesting to think about how that has changed. Current global estimates are that there are 300 to 350,000 individuals who've initiated PrEP. The majority of them are in my country, in the United States, but you can see the darkness of the red here reflects the number of people on PrEP, and that there are many global north countries, or Southern Hemisphere global north countries, who have a lot of PrEP use, but also some, as you can see, South Africa, Zimbabwe, and Kenya, standing out as countries that also have a fair number of people on PrEP already. What has changed? Some of it is messaging. and messaging that responds to what people care about and hear about PrEP. So these are smart and fun ads from the United States about taking PrEP. These are smart and sexy ads from Kenya about taking PrEP. I love the top one, just because the color matching is perfect. The PrEP and her lipstick and her nails, all perfect. And truthful ads. This is from Melbourne. Okay. I talked a little bit before about what people respond to about PrEP. Normalization has also been a really important thing for PrEP. So this is a Grindr screenshot, not mine, Grindr screenshot, but that says, do you know what PrEP is? In the yellow, do you use it? And then in blue, everyone seems to be on it in the US. This is a really important point. Everyone is on it, normalization. Why do you ask? I heard it's been approved in Europe now, in the EU. So this is really important, this normalization of PrEP as something. I live in Seattle. I work many places, but I live in Seattle, and I knew PrEP had arrived in 2014 when I read this article. The Case for PrEP, or How I Learned to Stop Worrying and Love HIV-Positive Guys. So a 30-something-year-old gay man who had never had sex with an HIV-positive gay man, because of fear, who did once he was on PrEP. So the normalization of PrEP, to be able to say it's okay for you to be taking PrEP because other people are taking PrEP around you, either people you know or people you virtually know online, and the stigma breakdown that PrEP has offered, I think has been quite substantial. It interfaces really perfectly with U equals U, in my mind. Makes me think we almost need a P equals P. You know, PrEP equals protected, or PrEP equals powerful. So in Seattle, where I live, this is what knowledge and stigma reduction has looked like. So this is a survey that's done at Pride every year. The numbers start at 2009 and go through 2017. You can see a rapid and substantial increase in the fraction of men who have heard of PrEP, going from about 20% to nearly 90% in 2017, and a fraction who've ever taken PrEP also rising substantially. The dotted line on the bottom one of ever taking PrEP, it now lands at about between 35 and 40% of men who have a characteristics that Public Health Seattle King County says would put them at higher risk for HIV are taking PrEP right now. So 35 to 40% in Seattle about right now are taking PrEP. We, over the same period of time, are seeing a substantial reduction in new HIV infections in the red line in this graphic, a 42% drop in new infections since 2010. Drops in new infections have been seen in several other big cities. In San Francisco, a 50% reduction in new infections over the past decade. In Sydney, wonderful data reported at CROI this year, a 32% reduction in community-wide new infections in the context of a demonstration project of PrEP rollout. And in London, a substantial reduction in 2016. These are not because of PrEP alone. These are part of combination prevention with very good testing and treatment services in all of these places. But importantly, this is PrEP layered onto really good testing and treatment services. So PrEP is undoubtedly having some sort of effect here on top of what is already really good rollout of testing and treatment. So these things work together and synergize. And this is what they say in Sydney. So I was in Sydney a month ago. This is a poster on the street advertising PrEP. Also part of normalizing. Messaging that's getting out there that people see it just passing down the street the same way you'd see a Pepsi ad. Programmatic rollout is happening in many places, and these are just examples here from South Africa, from Brazil, and from Kenya. And I encourage everyone to come at 7 o'clock tomorrow morning to the Kenya PrEP rollout program of one of the satellite sessions because it's going to be an excellent discussion. Other countries as well, Eswatini, Lesotho, Thailand are also doing tremendous work in rolling out PrEP at scale. What I've heard in the last few days here on things that are working in implementation, and just as there's an aggregate across both satellites and posters and oral sessions, what's really important for PrEP is easy access, or in the treatment world, differentiated care models. Making PrEP easy to get with few labs and taking a general population approach, particularly in generalized epidemic settings, that approaches that reach widely so that it is not stigmatizing to get PrEP because PrEP is talked about as something that everyone may apply to. Having a variety of delivery settings, including integration, some very nice work on MCH and FP integration, youth-friendly services, community-based delivery, online delivery, and this morning at a satellite session, standard of care and prevention research. As well as confirmation, yet again, that both daily and on-demand dosing are both effective and workable and work different for different sorts of guys. So impact has shown us that PrEP is working. So you put it together. PrEP works, PrEP is workable, PrEP is working. But it's not enough. So these are all things that are not enough yet. There are goals. UNAIDS has set a goal, optimistic, 3 million persons on PrEP by 2020. We are less than 10% of the way, we are on the order of 10% of the way there. But we are not at all 10% of the way to 2020. We are much further than that. 2020 is very close. So setting goals and reaching goals, we have much to do. Expectations are challenging. What is success for PrEP in terms of uptake, in terms of adherence and retention, in terms of cost and cost effectiveness and impact and incidence? These are yet to be determined. And using an ART model, which is what many of us come out of and what many of us set as expectations, does not work. The expectations for uptake and continuation and adherence to ART are completely different than they are for PrEP, because people can initiate PrEP or choose not to initiate PrEP, can be on PrEP for a while or can be on PrEP for a shorter period of time. If they are MSM, can use PrEP on demand and thus don't need to use a pill every single day, and thus adherence is calculated differently. We need to have global conversations on what is enough. We talked about continuation. This is a slide made by Gilead Sciences that looks at pharmacy refills of PrEP in the United States beginning in 2012 and 2013 in the black and the gray at the bottom, and then in 14 through 17 in the colors at the top. And what was seen in the United States based on pharmacy refills, that initial PrEP continuation was very poor. So 6-month pharmacy refill on the order of 10 to 20% in 2012 and 2013, and then jumping and changing thereafter. My argument is this is what happened with normalization. When people put PrEP on their Grindr profiles in the United States, it was normalized and was de-shamed, and people took it. And so normalization in the community is important. Barriers are the opposite of that. Stigma about HIV risk, about HIV, about being at risk, and about antiretrovirals. Access at the policy level, the program level, and then who's paying. In my country, what insurance is paying. In other places, what public health policies put PrEP into place. And then knowledge through marketing, community messaging, and normalization. Just to reiterate it, awareness. This is intentionally blank because this is what most policymakers, providers, potential PrEP patients, and all the people around them know about PrEP in the world right now, is nothing. And for a young woman in South Africa who takes home a PrEP bottle and asks her auntie whether she should take this or not, and someone knows nothing about PrEP and is told you have HIV or you are a slut or you are being poisoned, that does not support PrEP taking. And so normalization and messaging that's not just clinic-based but is population-based must make a difference. Complexity is not enough because PrEP has been framed in many ways as treatment, but PrEP is primary prevention. Someone this week said PrEP is a lifestyle choice. In many ways, it's analogous to contraception, so easier strategies seem to be necessary and seem to be really workable. Streamlined access, same-day start, integrated services, peer engagement, even peer delivery is being tried in some places. Minimize laboratory testing and community-based delivery. STIs are a complicated discussion on PrEP. This is data from Seattle. I showed more Seattle data before you. You know that HIV has been falling in Seattle over the last number of years, but STIs have been increasing a lot. Big numbers there. So I work on STI. I'm an STI researcher in the same way I'm an HIV researcher. There's a lot to discuss about STIs. What are needed are better diagnostics, active screening and treatment. In many places in the world, sadly, access to STI medications, even themselves, and new strategies for preventing STIs. At the same time, I remind myself all the time that we are in the business of preventing disease, not preventing sex. And we need to work from people where they are. So we need to find strategies to prevent STIs. I think about disparities in PrEP. This is data from the CDC. 44% of people who could potentially benefit from PrEP are African American, but 1% of those who have prescriptions are. And when I look again at that global picture of PrEP, it sits in it concentrates into richer countries, and in those richer countries into richer cities. And while I am happy that my brethren in Seattle are benefiting from PrEP, that's not the only people who should be benefiting from PrEP. And people need choice. And I'm encouraged that we have on the way, potentially, and we'll hear more about these from Naranzo soon, the potential choices that may be coming in PrEP. For contraception, women regularly can weigh side effects, efficacy, life burden, medicalization when making choices. And people want options so they can make choices. And I even include here on the right side vaccines and antibodies that are kind of analogous to vaccines, because the same sort of delivery strategies that will be necessary to give someone an injection every quarter for a year and boosters thereafter will be the same strategies that we need to capitalize on for PrEP delivery. It's not going to be any different. So we talked about efficacy, effectiveness, and impact. Is that going to be enough? I really do hope so. And I leave you with a great quote from Tony Fauci from the NIH, which he did in an interview in JAMA this month. He was asked, what's the most important progress we've made this decade in the epidemic? And his answer, treatment as prevention and PrEP, because if we really implement them properly, we can shut off the epidemic. I couldn't say it any better. Thank you all.
Thank you very much, Jared. So before you leave the podium, and we're going to open up your session for some 5 minutes of questions, I think, could you speak a little bit to the tension that seems to be arising between the traditionally HIV prevention intervention and the use of PrEP? For example, the issue of condoms, and we know that healthcare workers are probably going to consistently, insist on you have to use PrEP and you have to use condoms. Just from your experience, what do you think?
I can speak from my own clinical experience as a prescriber and from research experience. Yes,
not your personal experience.
Yeah, yeah. We'll set that aside. So PrEP, I take it back a little bit. So take it back for 1 second. The individual who most benefits from PrEP is the one who's already struggling to use condoms. PrEP doesn't cause people to not use condoms. PrEP is a backup when condoms are hard to use, either because because one can't enforce that, one can't remember that, or one doesn't want to. And individuals and isn't that what we're all about? We're all about giving people the tools they need to keep themselves safe. It is a hard one. It is a hard one. I'm a provider also. It's a hard one to say. It's a hard one to say you equals you. It's been hard to say you equals you. I think many providers would say that. It's been hard to say when someone asks you, do I have to use condoms now that I'm taking PrEP? But it's important to say, it's hard to say those in the same way it's hard to say you equals you. But it's important to give people the right kind of information so that they are able to protect themselves and not to scold people so that you can have a real therapeutic relationship. On the research side, we have a poster here. We have a poster here from Kenya talking about how hard it is for providers to say, I know in my head that individuals are virtually protected when they're using PrEP or when they're using treatment, but it can't get out of my mouth sometimes. But it gets better the more that we practice saying it. And how clients say, I don't understand why you say this PrEP is good if you say I have to use condoms every time on top of it. So either then I don't have an honest therapeutic relationship with you, or I don't trust what you're saying.
All right, thank you. So we'll open up for 5 minutes of questions. So if you have a question, if you can walk to the microphones, and then we'll take the questions. Please go ahead.
Hi. Yeah, great talk. I just wanted to ask, because when you show statistics from San Francisco and Seattle and maybe even London, you have the situation where you have a lot of poor people who have been moved out of those city centers. And so is there any example where you have a situation where say a Mississippi or, a place where the population has not been quite so mobile, where you've seen the same kind of drop?
I think I would have at least two responses to that. The first one is Sydney. The data from Sydney this last year from Epic New South Wales, in a very short time period, a very quick rollout of PrEP and a huge population drop in HIV, so not enough time that people would move out. And really, really good surveillance to be able to measure that. At the same time, you mentioned Mississippi. I wish that there was enough PrEP delivery at scale in Mississippi to be able to measure something like this. And that's what in Seattle, in in Seattle and in Greater Seattle, in Washington, D.C., in Greater Washington, D.C., in San Francisco, and the Bay Area around it, the impetus is very strong to shine a light on disparities and then address them through HIV prevention options like PrEP.
So thank you very much, Jared.
As we well know that PrEP works greatly for those who can actually use it, we also know that many people struggle with adherence and are not able to use it as needed. So we need to go forward and try to develop other options for people who are in need of it. This is also our mission. So we will have now Nirasdo Magodi, who will talk to us about what's new in the pipeline for PrEP. Nirasdo is a clinical pathologist working in sub-Saharan Africa, and she is the lead investigator in multiple clinical trials assessing biomedical HIV prevention intervention, like vaginal rings, oral PrEP, and also injectable PrEP, and studies with broadly neutralizing antibodies. So, Nirasdo, please.
Thank you very much, Beatrice, and thank you to the conference chairs and conference secretariat for inviting me to speak at this conference. So I've just spent just over an hour cutting my presentation, only to discover that I've got extra time. So I normally speak very fast, so I'm going to try to speak slower than I normally do. One of my bosses who's sitting towards the front always asked me to cut on my slides, and today I'm going to blame him. But anyway, we'll proceed. My presentation is entitled Next Generation HIV Prevention, What's New in the Pipeline for PrEP. I have no conflicts of interest to declare. So over the past 20 to 30 years, there's been a development of a wide array of HIV prevention modalities, and these have ranged from behavioral through structural to biomedical. And these have been in an unexposed participants or unexposed clients through to exposed and through to infected, where treatment has been used as prevention. My talk today is I'm going to limit my talk today to the pre-exposed for precoital and coital prevention of HIV, and I'm going to talk mainly about antiretroviral-based pre-exposure prophylaxis, as well as microbicides and monoclonal antibodies. The outline of my presentation is as follows. I'll talk about the various modalities of pre-exposure prophylaxis, and I'm going to finish off with a summary and a couple of recommendations. So we've heard from Jared that PrEP works if it's taken. And we all know, I think most of us were in Vienna we all know that PrEP works, and we know that in July of 2012, on the 16th of July, Truvada, TDF and FTC was approved by the FDA for use as prevention, and we know that success depends on adherence. Jared is going to task about this, but we know that adherence is effectiveness is related to adherence, and we know that the relationship between effectiveness and adherence approximates a sigmoid dose response curve. So the higher the adherence, the higher the effectiveness. And Jared has just shown this very well just now. We also know that success on PrEP depends on access. Yesterday, a colleague from South Africa talked about the missed opportunities for offering people for PrEP. There have been so many people who could have benefited from oral PrEP who didn't have the opportunity. And we know that out of the about 320 people who are on PrEP, about 320,000 people who are on PrEP, about 200,000 of them are in the US, as was said by Jared. In my country, about just 5,000 people are on PrEP. So already you can see that success on PrEP also depends on access. Some countries have not yet started oral PrEP. And besides adherence and access, some people might not prefer oral PrEP. The VOICE-D study looked at preferences for pre-exposure prophylaxis, and the women who were interviewed in the VOICE-D study reported that some of them would like injectables for pre-exposure prophylaxis. Some reported that they would like implants for pre-exposure prophylaxis, and then others talked about vaginal rings, and a percentage also said they would like oral tablets and the various forms of delivery of pre-exposure prophylaxis. So when it comes to preferences, we are mainly concerned about the end user. What does she want or what does he want? But it's not just about the end user only. We heard yesterday about how PrEP or how HIV risk is intimately related to intimate partner violence. So our end users might prefer something which is discreet, which is easy to use. And for women, especially women in sub-Saharan Africa, they might want something which is woman-initiated and controlled and does not need the consent of a male partner. What could this something be? This can come in the form of a dapivirine vaginal ring. These are long-acting PrEP-formulated rings which are formulated as flexible silicone rings that slowly release the antiretroviral dapivirine, which is an NNRTI with activity against many HIV subtypes. So the advantages of the vaginal ring is that it's got a potential for better adherence, it's long-acting, and it's got a strong safety profile. Like I said before, it's discreet and can be controlled by the woman. It's easy to use, comfortable, and does not interrupt sex. Most of our end users have reported that they want sex to be spontaneous, so a product that doesn't interrupt sex, exercise, or any daily activities is more likely to be favorable. And the dapivirine ring is scalable with a low manufacturing cost and can be stored at room temperature. Even these temperatures of 34 degrees Celsius in Amsterdam, I'm sure the ring can survive. Right, so the dapivirine ring has been assessed in 2 phase 3 randomized clinical trials, the ASPIRE study as well as the RING study, where over 4,500 women aged between 18 and 45 were enrolled in the study, and they used the ring monthly with replacement of the ring every 28 to 35 days. What these 2 studies showed is that the ring is safe, is tolerated, and it can reduce the risk of HIV by about 30%. So after these 2 phase 3 randomized clinical trials, the researchers went on to perform 2 phase 3B open label extension studies to the ASPIRE study and the RING study, namely the DREAM study and the HOPE study. So women who had participated in the ASPIRE study and in the RING study were offered open access to the dapivirine vaginal ring. These 2 open label extension studies showed that increased adherence, showed that with open label use, there was increased adherence, and there was also sustained safety profile of the ring. And comparisons of HIV incidence using statistical modeling suggested HIV risk was reduced by 54% in both of these studies. So looking at the phase 3 studies, risk was reduced by about 30%, and in these open label studies, risk was reduced by 54%. So where are we at with the dapivirine vaginal ring? Currently, the dapivirine vaginal ring is being reviewed by the European Medicines Agency under Article 58 application. Should EMA provide a positive opinion, then the International Partnership for Microbicides is going to seek WHO pre-qualification. And with WHO pre-qualification, there will be submissions to other especially African regulatory agencies, for example, in Kenya, Malawi, Uganda, and my country, Zimbabwe. So already there's a lot going on in terms of the vaginal rings. So while we wait for the EMA assessment, what has been going on? Ongoing and completed studies are currently evaluating the dapivirine vaginal ring among adolescents, among pregnant women, among lactating women, and among women who are aged 45 years and older. Already there's development of next phase products with the looking at longer acting rings. Remember that dapivirine currently, the ring currently lasts for a month, but now there's development of a 3-month product, which is in phase 1. This is going to increase convenience and, of course, lower yearly costs. There's also development of multi-purpose rings. For example, there's currently a ring with levonorgestrel and dapivirine, which is in phase 1 studies. This, of course, will target pregnancy and HIV protection together. We are also preparing for delivery of the ring, where currently there are plans for side-by-side comparisons of use and preferences for oral pre-exposure prophylaxis and dapivirine ring. And ideally, demonstration studies and operations research to understand real-world delivery are already in place. So with that, I have no doubt that we are poised for success. And please allow me to thank our funders and the women, the researchers, the research communities, and the women, the thousands of women who've taken part in the studies. But also allow me to thank the beautiful and heroic women of the mythical African country of Wakanda. These women have not only shown us that, yes, it is okay for Black women to have coarse hair, to have braids, to be bald, or to be serious. These women have shown us that it is okay for us to be dark-skinned. We don't have to bleach our skins to be beautiful. They've also shown us that when we are faced with adversaries, the wise will break barriers and build bridges. So as such, because of these beautiful women and the beautiful women of Africa who've taken part in these studies, we are set to have an HIV prevention product which might look like this, whether it's working against an enemy in Wakanda or it's working against HIV in African women. But they've also taught us about other weapons which can be in the shape of spears, which resemble long-acting injectable products. So as I move on to long-acting injectable PrEP in HIV prevention, it's got many advantages. We've talked about adherence, and Jared has also talked about adherence. So the advantages of long-acting injectable PrEP in HIV prevention is that injections could be given every 1 to 3 months, and this could address adherence issues, and it's not event-driven, so adherence is more likely to be better. Also, for long-acting injectable pre-exposure prophylaxis, we'll be using different drugs, not used heavily for treatment. Therefore, there is less concern for resistance and cross-resistance. Also, what has come up from our stakeholder consultations with communities is that people are familiar with injections, and these are highly acceptable, learning from contraceptives. And also women have talked about how injections are more discreet and more private than pills or rings. So these are some of the advantages of long-acting injectable PrEP. Coming back to tenofovir-based pre-exposure prophylaxis, one limitation, which sometimes we don't really talk about, is the competition by deoxyadenosine triphosphate, DATP, with tenofovir diphosphate for binding to reverse transcriptase. So once there's that competition, there's going to be possible weakening of tenofovir-based PrEP protection by inflammation. And we know that inflammation leads to T cell activation, which leads to increases in DATP, and increases in DATP are going to large amounts of DATP are going to compete with tenofovir diphosphate for binding to reverse transcriptase. So there is weakening of the protection by tenofovir-based PrEP. So let's look at products which act directly on the viral protein, which might circumvent this challenge. Products like cabotegravir. Cabotegravir is in the long-acting formulation. It's got favorable attributes for PrEP. It's got a high genetic barrier to resistance, and it's got a favorable PK profile with a half-life of 21 to 50 days, and it allows once daily. It allows once daily oral or 1 to 3 month injectable dosing using a nanosuspension formulation. So currently, to date, a number of studies looking at long-acting cabotegravir have been done, and it has been shown to be safe and effective. So I'm going to talk a bit about HPTN-084. This is a study which is evaluating the relative safety and efficacy of cabotegravir versus oral FTC-TDF for HIV prevention in women. This is a phase 3 superiority trial which is comparing once daily oral PrEP, oral Truvada, to oral or injectable cabotegravir. It's a randomized, double-blind, 1-to-1 double dummy study where all participants receive one active and one inactive product. Because once the injection has been given, it cannot be removed even by dialysis. There's need for an oral run-in period of 5 weeks where we confirm the tolerance before participant is moved on to long-acting injection. After the 5 weeks of oral run-in, there's the injectable phase where participants will receive either injectable cabotegravir with placebo oral Truvada or placebo injection and oral Truvada. So this step 2, which is injectable, will last up to 185 weeks. And after that, there will be 48 weeks of open label use of Truvada to cover the tail phase. So this study is being conducted at 20 sites in 7 countries in sub-Saharan Africa. And my apologies there to the Swazi. You're supposed to read one site in Eswatini, not one site in Swaziland. But welcome to the newest kingdom there. The study is looking to enroll 3,200 women. And the study opened in November of 2017, and to date, 6 sites have been activated. And as of yesterday, we had enrolled a total of 155 women who are at risk for HIV, and retention remains high with greater than 92% for all visit types, including the safety visits as well as the injection visits. So there are already other considerations for HPTN-084. HPTN-084 will assess the decay curve for women who become pregnant. The trial will assess the decay curve for CAB levels in a pregnant population and compare with non-pregnant women. We'll also look at the effect of drug exposure at the time of approximate time of conception on infant outcomes. We'll also characterize the washout of cabotegravir concentrations in a subset of infants and their mothers. This will be done in the breast milk and infant PK study. There's also a planned contraceptive sub-study where we'll determine plasma concentrations of medroxyprogesterone or noethistron or etonogestrel when co-administered with CAB-LA, and we'll look at the drug-drug interactions. So I've talked about the advantages of long-acting injectable. I would like to acknowledge Ian McGowan for this picture. It's not the exact one he used, but this picture is just to illustrate a Komodo dragon, but also to remind you that with injectable, long-acting injectable, there's a long tail phase. So with this long tail phase, there's safety and resistance issues and especially if a participant becomes HIV infected. I've also mentioned that once the injection has been given, it cannot be removed in the case of adverse events or when there's desire to stop PrEP. So all these disadvantages point us towards a product that can be removed and would not need an oral lead-in phase or cover for the tail phase, because once it has been removed, the drug effects would also be abolished. So we need a product that could be removed. Products like subcutaneous PrEP implants. The use of implants has been there for quite a long time, especially in hormonal contraception, as well as the management of hormonally driven malignancies. So already we've got a lot of experience in the use of PrEP implants. And the advantages of PrEP implants is that they could have consistent and predictable drug release, and they could be longer acting compared to injectable and may remain in place for up to 5 years, and they could be inserted and removed simply. If they are radio-opaque, they can be tracked using radiography, and we could have co-formulations of PrEP and contraception. But they don't come without disadvantages. The insertion and removal would require a sterile procedure. And I said that the implants can be palpated, but palpating doesn't mean that the implant is active. So they might be expired in situ implants, or they might lead to scarring or tenderness at the insertion site. So currently there are a number of implants which are under development. There's TAF, there's CAB, there's EFDA, which is almost the most advanced. So we have a number of products which are in development for PrEP implants. And colleagues from the Northwestern University running a program known as SLAP HIV, Sustained Long-Acting Protection Against HIV. And what they are looking at, the major question they are asking is, can we develop an implant that can protect, that can provide protective art for a year? So moving from PrEP implants, we are going to talk about antibody protection. So in terms of passive antibody protection, I'll introduce 2 phase 2B efficacy studies known as the AMP studies, which are trying to answer the question, can a passively infused monoclonal antibody prevent HIV infection in high-risk adults? These studies are run as 2 harmonized cohorts. The first cohort is looking at MSM and transgender people in the North and South Americas, as well as Switzerland. And the other cohort is looking at 1,900 heterosexual women in 7 sub-Saharan African countries. These are placebo-controlled proof-of-concept studies of VRC01, a monoclonal antibody, which is given intravenously on a 2-monthly schedule. Both trials opened in April and May of 2016, and to date, both trials are nearly fully enrolled. And the study has progressed well, with retention at 96% and more than 99% adherence to infusions. So what is next in terms of broadly neutralizing antibodies? A new generation currently of highly potent and broadly neutralizing HIV antibodies has been identified. And such antibodies can be modified to improve their potency, their breadth, and the half-life. And this can be done by genetic engineering and is going to optimize the chances of success. We all know that HIV exhibits genetic diversity and viral escape mechanisms. So it is prudent for us to consider using a combination approach, and we also need to learn from art that combinations of antiretroviral therapy could also help. If we combine monoclonal antibodies, we could reduce the likelihood of viral escape and increase neutralizing breadth. So we know that combinations of broadly neutralizing antibodies and trispecific antibodies can bind to virions and prevent HIV mucosal infection. So infection can be prevented in the mucosa, or they can elicit, the antibodies can elicit antiviral responses deeper in the tissue. It is also hoped that using this multi-target approach could prevent resistant breakthrough. So with this in mind, already there are next generation broadly neutralizing antibody studies looking at safety, PK, PD, and potential. The HVTN and the HPTN are working on several studies. One of them is looking at long-acting VRC07, the long-acting variant of VRC01. There's also a study which is going to look at the Sanofi antibody, which is a highly potent and broadly neutralizing trispecific antibody. And also there's going to be trispecific antibodies which are going to be assessed. So already there are many studies which are looking at next generation broadly neutralizing antibodies. So in summary, looking at improved delivery methods to allow for long-acting products, this may prove revolutionary. When we complete the current studies that we are doing, we should have a long-acting PrEP agent. We should also have advanced broadly neutralizing antibodies for prevention of HIV infection. But we must learn from history and recent findings. There's been a lot of talk about dolutegravir, and there's a lot of anxiety surrounding dolutegravir. So it is recommended that we should have concurrent or as near concurrent as possible data collection of pregnancy and lactation safety. For oral PrEP, data are still being collected. For dapivirine and CAB-LA, I'm glad to say that there are good research plans to get this information. And for dolutegravir itself, we should gather as much information as possible. We should also institute lessons learned from oral PrEP. To quote Jared, who is seated here, he said, "We must learn from oral PrEP implementation to catalyze implementation of other prevention tools, like the implants that I talked about, the monoclonal antibodies, and the injectables." Michelle Warren yesterday talked about other lessons learned from oral PrEP, that post-approval studies were not all designed to address decision-maker questions. And data from research was not well-timed to inform decision-making at global or country level. And also at times, the stakeholder landscape was complex and fragmented. So we must learn from these lessons so that we don't repeat the same mistakes. And we must remember that the approval pathway for new HIV prevention drugs could be more complex than for a drug already approved for treatment. So with that, I would like to acknowledge a whole team of investigators and colleagues who assisted me with this slide set. And I would also like, on behalf of the co-chairs and the chairs, to remind you about HIV R4P and to say that I hope to meet you in Madrid. Thank you very much.
No questions from the audience? So I can start. So Niraz, do you show high levels of adherence and retention to the monthly infusions in the AMP study? Did you do any qualitative studies to understand what's behind this, what's moving women to come? I know the men are also coming as well. Do you have any information on that?
So when we started the AMP study, we were a bit worried about whether there will be a good uptake of the infusions. So to date, we've had an adherence rate of over 99%, and I think we owe this to the extensive community engagement that took place before launching the study, during the study, and as we implement the study. So there's a lot of community sensitization, but we do not have a qualitative component of the study, but we do have anecdotal data as to why women prefer this the infusion.
Thank you. Please.
I'm Karen Beckerman from New York City. I work in the South Bronx, and I provide prenatal care for women at risk in general because of the endemic nature of HIV in our community. We do not suffer from lack of PrEP, and certainly ID specialists and many obstetricians are aware of its availability. But the block that we cannot seem to overcome this year is lack of knowledge of women at being themselves at risk, and also lack of knowledge among colleagues in other subspecialties or other specialties. I don't really see how these exciting data that you've presented address that at all. How are we going to get partners of our clients to test? And what studies are out there to encourage not just testing of, say, a pregnant woman or an individual presenting to an STI clinic, but to encourage testing of couples together? Thank you very much.
Thank you. Thank you very much for that question. And I'll start to answer it, and I'll also call on Jared to assist on the other studies. I totally agree that there is lack of information to some colleagues in the health sector. So we need to have community engagement to encourage or to disseminate information to healthcare providers so that they encourage couples testing, and that women know their risk status as well as the status of their partners. So Jared, do you know any studies where we are promoting testing in couples?
Yeah, I think testing of couples are offering. There's been a lot of work done to think about offering self-tests to women to take back to partners, not so much in the United States, but in higher burden areas. I think also a lot of discussion about recognition of risk, or at least recognition of PrEP, and that for PrEP, that's a tension whether someone has to declare that they're at risk or just declare that they want PrEP. And as PrEP normalizes in delivery settings and providers are more comfortable about offering it, people can just say, I want PrEP, without having to it's hard for anyone to declare that they're at risk of anything. I would have a hard time declaring I'm at risk of having a heart attack as a approaching middle-aged American man.
So perhaps just to add on, in terms of risk, discussions at AIDS 2018 have shown that the perception of risk is a major driver for the uptake as well as the continuation of PrEP. So we really need end users to really look at themselves and to determine their risk. But even though they don't see themselves at risk, we should offer them PrEP as much as possible. Yes.
So please.
Good afternoon. Cassidy Clausen from University of Maryland, based in Zambia. And thank you so much for the two presentations so far. My question is for both presenters. In our work implementing PrEP in Zambia, we've found a fairly rapid drop-off in adherence or PrEP persistency, as is common, as is seen in South Africa and other places. So by month 3, we have 23% who remain engaged in care. I think the question that is really out there for a lot of women in the African setting is a question around on-demand PrEP. IPER-GAY trial, of course, demonstrated on-demand working well for MSM, but the current recommendation is that it takes 21 days to reach therapeutic levels in the vaginal tissue. So my question comes then to dalpivirine ring, where we're seeing only 30 to 50% efficacy. Do you think there's the future of PrEP for women will be a combination approach, where we take a long-acting injectable or implantable or ring that is paired with something that then is more on-demand based to improve efficacy, but also improve utilization of the therapeutic approaches when they're available? Thanks.
Okay, so I'll start, and then Jared, you can pitch in. So in terms of the efficacy of the dalpivirine ring, the intention to treat analysis for the two studies, ASPIRE and the ring study, showed a reduction of about 30%. But what we know is with more improved adherence, if adherence was as high as 80%, then efficacy also increased higher, up to the magnitude of 80%. So with the ring, I'm quite hopeful that there will be higher protection, because adherence with open use, with open label use as well as real life use, we expect adherence to be higher. So in terms of a combination of on-demand and the ring, Jared?
So the hard one. I think that a few things that I think about a lot. I think it really emphasizes how important choices are. That individuals have to figure out things that work. That we have to keep working on prevention products that will work for different people at different times of their lives. One thing that I think we all left out of our presentations today is this: there's probably still a need out there for an on-demand. We talk a lot about long-acting. There's still going to be a need for an on-demand PrEP option, including a PrEP option that women can use, and we have to get the research to develop all of that. I would also say the big drop-offs that we're seeing in oral PrEP was very similar to that slide I showed from the U.S. That we saw the same thing in the U.S. and then we saw a change in time. And I think we also have to think about the ways to normalize the discussion that PrEP exists in PrEP takers, their providers, but also the people around them, the peers and the parents and the partners around them, so when someone leaves the clinic, they're not totally alone.
Yeah, so I think we'll move on to our next presentation, and hopefully we have time to answer a few questions towards the end. So I want to introduce David, who's our next speaker, who's going to talk to us about controversies of designing trials of new prevention interventions. David is a professor of biostatistics at the University of California, San Francisco, and has worked in the field of HIV treatment and prevention for more than 25 years. And in the last decade has worked on pre-exposure prophylaxis and was the protocol statistician for the IPREX prior. So David, please go on.
Thank you very much. So yes, up front with my disclosures. Wait. Good thing we have plenty of time. No, that's the wrong presentation. So up front with my disclosures, I'm terrible with PowerPoint. I did a site visit going backward once. And I serve on an advisory board for Gilead Sciences, and I'm a practitioner of the art of statistics. I'm going to talk to you about an issue that has some technical components to it, but it's one that I've been really passionate about lately. Because what I care about is how these great products that we can conceive of actually can make their way into use. True that no one started a product because of a p-value, but some p-values are necessary along the way, which is why I have a job. I'm going to do it. And so my idea here is I'm going to talk about the standards and the analyses that we might do of future trials of novel PrEP agents. I'm going to be particularly interested in the cases of antiretroviral-based PrEP that is non-oral. So I'm very interested in the implants and the injectables. And I'm not here to discuss other people's controversies. I'm here hopefully to say some controversial things myself. If we're going to talk about studies of injectables and implants, we should acknowledge that these studies are already in the field. You heard about HPTN 084. I'm a little more fixated on HPTN 083. This will enroll an HIV negative population of MSM with at least 10% representation of transgender women, and it will randomize those participants to either receive cabotegravir as an injectable or daily oral Truvada. You heard about the run-in phase and you heard about the tail phase, but an important aspect of this is that it's a double-blind, double-dummy study. So all the participants will take pills and receive injections, but half of those will be placebo and half of those will be active. This is Beatrice is the co-chair of the study, so naturally, I think it's amazing. I think it's an incredible effort on the part of the HBTN. It's a very rigorously designed study. And I have only one concern about the design of this study, and that is it is absolutely enormous. So let's pause again. 4,500 participants to be followed for an average of 2.5 person years. I want to put that in some context for you, because it's incredibly important as far as the issue that I care about, which is how do we move these products along? And what I'm showing you here is the planned person years of follow-up of 2 of the MSM future PrEP trials, and compare it to what was achieved in the IPREC study as a part of its primary analysis. Okay? All 3 of these trials in an MSM transgender women population, all of them assessing a PrEP product. TDF-FTC in the case of IPREX, which is completed, and 083 and the Gilead-sponsored Discover trial are assessing injectable cabotegravir and oral TAF-FTC. Look how much bigger. Even though we're conceptually getting at the same thing, same population, these are about 3 times the person years, which roughly maps to the cost, the time, and the expense of these studies. So this becomes a formidable barrier to advancing new products. We heard about 3 separate things that could be implants. How are we going to test all of them if we have a pipeline that requires such large trials? So my fixation became understanding why trials like this are so much bigger. It has to do with the fact that they're designed as non-inferiority studies, but that's just on the surface. The way that they're built and powered relies on an assumption and a standard, and I have a problem with both of them. So I'm going to start with the assumption. These are a generation of active controlled studies. So we randomize people either to receive daily oral Truvada or, let's say, long-acting cabotegravir. And what I have here is fake data of the kind of results we might see at the end of HBTN 083. That study plans to observe 174 seroconversions. That's what it's planned for. And what happens is when you have an active controlled trial like this, you can compare the seroconversion rates, 2.0 on oral TDF, 1.9 on injectable cabotegravir, and see how they compare to each other. But you may remember from the first generation of PrEP trials how variable the effectiveness of daily oral TDF-FTC was. So you're able to compare to a standard, but that standard is highly variable across contexts. And so we can say that they're similar, but are they similarly effective, moderately effective, or highly effective? And the way that this is disambiguated is by introducing an assumption. That's the assumption I'm going to complain about now. So these trials, Discover and 083, as non-inferiority trials, use a method called 95-50 to power them. There's 95, there's a 50. The 95 part is an assumption, and it makes an assumption about how effective on an intention to treat basis oral TDF-FTC will be in 083. And it bases that on performing a pooled analysis of the placebo-controlled trials in a similar population that were done comparing TDF-FTC to placebo. So we really have 3 trials that somewhat meet that definition in an MSM population. When you perform that meta-analysis, you take a skeptical value, and that skeptical value is the lower bound of the 95% confidence interval, which in this case is 34% efficacy. So that's quite a skeptical idea of the effectiveness that we will see in 083. It's smaller than we saw in any of the trials to date. And we use that then as the benchmark for understanding how well cabotegravir did. Because cabotegravir did slightly better on the previous slide than oral TDF-FTC, it translates to, under the assumption of 34% efficacy for TDF-FTC, we get 38% efficacy for cabotegravir. So this assumption comes from entirely external data. Now, here's an additional part of the problem. There are not going to be any more placebo-controlled trials. So no matter what the product or what the new study is going to be, for MSM, we're always going to be dipping back to the same fixed set of data, and it's going to be returning this very skeptical value for the effectiveness of TDF-FTC. Okay. But I also don't like the standard. And in fact, this may be more of a problem. These studies are formulated as non-inferiority studies. And they define non-inferiority of one product relative to the standard as retention of at least 50% of the effect. This is a criterion only a statistician could love. Truly. And I don't even like it. The problem with it is that it sounds very reasonable. Who wants to give up effectiveness, right? If we have a very effective product now, why would we want to give that up with a new product? But the nature of how this is formulated statistically is it operationalized as a higher standard. So Snap-in showed that products that would normally meet the standard that TDF-FTC met, which is just to show it's effective relative to no PrEP, that's actually a lower bar than a 50% effect retention. So if the question is, why are the trials so much bigger now? It's because we're demanding more of the new products. Okay. But here's the thing. You can articulate a standard like this, but what is the rationale behind it? What is the thinking behind it? The thinking behind this, as best I can tell, is that we would certainly not want to go backwards if we were to replace Truvada with a new product. But when we look at the context of PrEP that we're in, with so many people needing prevention, not on Truvada, we're not seeking new products to get our Truvada users to take something else. What we're seeking are options for people who can't or won't take Truvada now. So as I say, we're seeking an alternative, not a replacement for Truvada. And that makes this standard out of touch with what we need. There's nothing in the standard that says anything about choice. And we've heard a lot about choice when thinking about new options. I want to go back to banging on the assumption now. So here I'm showing how we would interpret the results of that hypothetical HPTN 083 data. And I'm showing what we would conclude for that based on the full results of the meta-analysis, both the upper confidence interval in the green, the estimated value from the meta-analysis in the blue, as well as that highly skeptical value in the red. And you see that long-acting cabotegravir, we would interpret very differently its effectiveness based on the assumption that we make about the effectiveness of TDF-FTC. This assumption is critically important, okay? We really want to get it right. If we guess a value that is too high for TDF-FTC effectiveness, it makes the new product look better than it should. If we assume a value that is too low, we make the new product look worse than it is. And that is highly believable when we choose a deliberately skeptical value like 34%. So why not instead absorb the lessons that we learned in the first generation of PrEP trials? And that is, effectiveness varies because of adherence, and adherence is well measured by drug levels. So why not instead use pharmacology to give us a sense of adherence, use the relationship that we have learned between adherence and protection, and infer what the protection was in the trial? I do want to say that my colleagues who designed the HPTN 083 trial actually proposed a primary analysis based on doing this, but that was rebuffed by a regulatory agency, which shall remain nameless. I want to return now to the standard, because this is where I think we've really made a bad choice for our PrEP products, okay? So this 50% standard is all about substituting one product for another. But a real problem is the underutilization of highly effective prevention. We're not looking to switch current Truvada users, but instead to engage people who currently can't or won't take Truvada. So we should have a standard that reflects that. And this is what I contend. What we want from a new product is a new product that, when used as directed, is highly effective in preventing HIV. So a very efficacious product when used as we direct. And that such a product is also highly acceptable and actually used by people who don't or won't currently take TDF-FTC. So how do I see that going in a study? What I suggest the standard should be is performing as-treated analyses of the novel PrEP product in clinical trials. So looking at those as-treated analyses to get at the efficacy of this product. We're able to compare it to the background incidence because we back-calculated from drug levels. And what we want to show is that we can rule out low to moderate efficacy. And what we used in the first generation of PrEP trials was a straw man of 30% or lower. Fortunately, IPREX did not meet that standard, but that was the standard that was used to power those trials. And finally, we would want actual use data that showed that this product is used as directed by some significant portion of those who can't or won't take Truvada. So just to kind of put this in a framework for how I see a path forward for these types of studies, I believe that their designs would look very much like HPTN 083. They'd be double-blind, double-dummy studies of the novel product versus oral TDF-FTC. Pharmacology would be key for these studies. So we'd make sure that we're testing the drug levels, particularly on the TDF-FTC arm, possibly on the novel product arm. Although when we think about implants and injectables, these are generally a directly observed therapy. So we'll have a good handle on use without pharmacology as well. We use those drug levels on the TDF-FTC arm to infer the background incidence rate to disambiguate the trial. And we focus on proving that the novel product, when used as directed, reflected through an as-treated type analysis, can rule out low to moderate efficacy. Finally, one of the advantages of a double-blind, double-dummy design is that we can actually estimate the size of and the attributes of those who will adhere to the novel PrEP method but won't take Truvada. Because what we'll do is on the Truvada arm, we can test for pharmacology, identify non-users based on pharmacology, and look at the pattern of their receipt of injections or implants, and possibly relate that to participant characteristics. The important thing here is now that we're talking about a revealed preference, not just a stated preference. Because we know from the stated preferences that people really want injections and implants, but do the people who aren't taking Truvada, will they really come in for those modalities? So to tie it all together, we're in a situation where this approach that's been used to power the trials of next generation PrEP agents has led to enormous sample sizes. And this has incredible implications. Our resources are constrained, both in money and in site capacity and opportunity cost. We want to be able to rigorously test new methods for PrEP, but we want to be able to do so in a way that is more aligned with what we really want from these agents and what we've learned from the PrEP field. So I believe that we need to anchor our expectations for disambiguating the active controls using drug levels, and I think we really need to reconsider this non-inferiority standard, which creates large sample sizes and is not attuned to providing an additional option for people who want prevention. So I want to thank some colleagues that helped me think through these ideas, and I hope I've lit a fire under some of you to care about this very important issue that bears on how and when we'll have new PrEP agents available. Thank you.
Questions from the audience? I just wanted to make a quick comment. I won't dive into the statistical issues, of course, but just to let you know that we are half enrolled, doing spectacularly well, enrolling in a pace of 200 individuals a month. And that we also have, just a second, we also have very rigid metrics that we are following about enrolling the target populations that most struggle with oral PrEP. That transgender women, as you said, young individuals younger than 30 years old, and also Black MSM in the United States.
So there's an aspect to the design of OA3 that is trying to make preferentially select people who would struggle with oral adherence, in part to align what they expect to see with that meta-analysis. I can hear you, and I'll repeat the question.
Thank you. Maybe that's on now. Will Goodell, I'm a PhD student at Brown University. I had a question for you, Dr. Glidden, about maybe potentially using other sort of surrogate markers of risk for HIV risk as an endpoint in these trials.
So there's been a lot of discussion in the ambient world that thinks about these things, about other ways other than drug levels. I'm certainly open to that. What we know is that, let's say, to STI incidents, I'm just worried that as as prevention scales up, that historic relationships between STIs and HIV will kind of diverge over time. And so I'm a little cautious about extrapolating that.
Thank you.
All of this involves an assumption, and I'm more skeptical of that one.
Wakefield with the HIV Vaccine Trials Network. I know nothing about statistics except statisticians eat in the same lunchroom that I do. And I'll probably have to watch your presentation a couple of times to really understand my own question. But I didn't hear you exactly say whether we should be moving in terms of a new design or new policies around the engagement of statisticians in terms of design. And that's one question. And the second question was, you made an absolute statement that we will never again have placebo-controlled trials. And I wonder if that has been vetted with individuals who might enroll in trials, because I don't know that we've had the ethical debate about whether or not persons, again, have a choice of deciding whether or not they would like to enroll in a placebo-controlled trial.
Sure. Maybe I always try to strip absolutes from my talk as I speak. It's hard for me to foresee a situation in which there is a trial that is placebo-controlled, HIV endpoint-driven, of Truvada versus placebo. The question of whether or not there would ever be placebos in a trial, I mean, the word placebo can mean a lot of different things. There's cabotegravir placebos in 083 and Truvada placebos, but I find it hard to believe that we wouldn't at least offer PrEP in a future prevention study. What we have is we have the WHO recommendation that people who are at least 2% risk of HIV be offered PrEP. So I think the offer at least has to be made, and people have autonomy not to take PrEP, so they exercise regularly.
So we don't have time for any more questions, unfortunately. So I'm going to ask anybody who has any additional questions to speak individually to our speakers. But I want to thank all the speakers. Let's give a hand for the very interesting presentations. And also thank my co-chair, Beatrice. I hope that's the right pronunciation. And enjoy the evening. Thank you.